right, so thank you very much. Thank you for the organizers for putting together this wonderful conference. All right, before I get into my talk, a quick outline. I'm going to present some background. Then I'll look at kind of the main question and our main idea, and I'll give you a synthetic example that hopefully helps explain kind of what we did, and then I'll take some time going into the more mathematical parts of the story, and then I'll end up with an application to the motivating example. All right, so the project that I want to talk about today arose from this very interesting grant that I was part of. So this was funded by the American National Science Foundation and the Simons Foundation. And this project brought together 7 pairs of mathematicians and biologists who didn't know each other. So kind of new collaborations that were forced upon us, and they gave us money to do it. Okay, so for me, I was given funding for actually 2 postdocs, and my collaborator Hong Lu, who's a biologist at Georgia Tech, was given funding for graduate students. And our trainees were forced to talk to each other, and the PIs also had to talk to each other, and we had to figure out how to do something interesting and useful. And of course, working with a collaborator in another discipline, there's a big language barrier initially, and I know many of you in the audience have worked with collaborators outside of mathematics, and it's a real challenge understanding what scientists are trying to do, how they are approaching their big scientific questions, and then trying to come up with a way of how mathematicians can collaborate and contribute to the scientific endeavor. And hopefully also find something in the science that leads to interesting mathematics. So my collaborator Hong Lu, so she's a biologist for the purpose of this talk, but really she's an engineer. Her expertise is in microfluidics. So she has these little devices here, microfluidic devices, in the middle picture there, and that gives you kind of a sense of the scale of the, and you don't really see what's going on there. But inside there, what she's studying is this little worm, or nematode more precisely, C. elegans. It's at the very top. It's a very tiny organism. The scale is like a millimeter, or a couple millimeters, that's the length scale. And this is a very well-studied organism in biology for a number of reasons, one of which is it's very simple, but it's still somehow complicated enough that it's a proxy for things that we're really interested in, which is ourselves. Okay, so it does have a brain, it has a nervous system, but it's very simple. It has 302 neurons. Okay, so biologists have figured out exactly all of the neurons, and even they have a pretty good understanding of the connectome. And furthermore, this organism has been manipulated so that we can, in some sense, see them thinking. All right, so when a neuron activates, lots of physics, chemistry, biology happens, and there's a flow of calcium ions, and certain proteins have been engineered to phosphoresce in the response to these calcium ions, and these worms have been engineered to have these phosphorescent proteins in their brains, so that when they are thinking, a light flashes. So the neurons are activated, light flashes, and the other fantastic thing about this organism that I haven't told you is that they are translucent. Okay, so we can look at them with optical microscopes, just kind of standard microscopes, but fancier versions of them, and we can see and take videos of these worms thinking. All right, and because of these microfluidic devices, we can our collaborators can manipulate the worms and track them and see videos of them thinking in response to their environment. And the other reason the biologists really like these organisms is because they are very happy to live in lab conditions. So they are happy to go about their business on top of a gel in a petri dish, okay? Or within one of these microfluidic devices. All right, so on the bottom here, we see one of these videos that our collaborators captured, and you see some lights. If you look at the slightly better resolution on my laptop screen, you see the white spots here are not constant. They're getting slightly brighter and dimmer. And then through various, a lot of work, we get kind of the picture on the right here. Okay, so let me say a little bit more about that. So one of Hong's students, Shivesh Chaudhuri, spent a large chunk of his PhD producing this one image right here. Okay, so of course he did a lot more than that as well, but this was kind of the highlight for us. Is this is a matrix, okay, and it's rows and columns of real values, 278 rows, sorry, 278 columns and 72 rows. Each row is one neuron that's been identified in each of the frames across this video, and its activation has been measured for 1 second of this 278 second video. Okay, so we have this, this is kind of a heat map slide here in the bottom left, and zooming in on about a dozen of the rows here, and then connecting all the 278 dots, we have these time series. Okay, so this is a discrete time series, 72 neurons, 278 seconds worth of video. And we're seeing the worm thinking. All right, so this is the type of data that they have, and what are they interested in? Of course, they want to understand how brains work, and they're working with very simple organisms, but they have big questions. One big question is, how do brains learn? Okay, how does that happen at the neuronal level? So let me give you an example of an experiment that the biologists would do. Is they can feed these worms. The nematodes eat E. coli, and they react to their environment. They're sensitive to smell, for example, so they smell E. coli and then they look around for it. And now, but if they, now biologists are mean, and they're allowed to do mean things to worms because nobody cares about worms. So they feed the worms E. coli that is poisonous. All right, and most of the worms die, but those that survive, the next time they smell that E. coli, they're going to react differently. So a big question is, can we look at the data and somehow try to unravel that? Maybe hopefully using mathematics, topology, to say something about the learning process. So of course, I'm not going to tell you that we solved this problem. This is kind of a big problem, but what we wanted to do is develop mathematical techniques using topology that would hopefully tell the biologists something useful, and maybe allow them to look at their data and have hypotheses for further experiments. All right, so that's what we tried to do. And so of course, in applied topology and topological data analysis, we have a standard method for looking at time series data, and that's the sliding window embedding. And here I have a very toy example. If you haven't seen sliding windows embeddings before, this is the simplest example. We have a simple time series, just a cosine curve evaluated at a bunch of points. As a time series, this is just one-dimensional data. And of course, in topology, to see periodicity, we want to see circles. So we take a higher dimensional embedding of this one-dimensional data. We embed one-dimensional data into 2 dimensions by taking consecutive time points of the cosine signal, and that of course gives us an ellipse. All right, so that's what we see here. All right, now in our example we have 72 neurons. Here's an example with 5 signals, and there's 2 ways we can go about doing sliding windows embeddings. And the first of which is to look at each of these signals individually. Okay, so for some window length, we collect all of those points, and then we get a point in R to the L. L is the window length. And as we sweep across the time series, we get a point cloud for each of these signals. Okay, so I'm going to call this the component embedding, one for each of the components in the signal in the time series. And this is not going to be the main thing that we're going to analyze. Instead, we're going to look at what I'm going to call the global embedding, which is that we look at the multivariate time series. We look at kind of all of the values, all the vector values in that time series. And now instead of kind of having L numbers in the window, we have L times, in this case, 5 L numbers. So as we sweep across, we get a time series embedding in 5L dimensional Euclidean space. All right. Okay. And I didn't say this at the beginning of the talk, but please interrupt me throughout my talk. I know there's a lot of students here, and of course you can never cover everything in the lead-up workshop. So if there's anything that's not clear, please stop me. I'm happy to answer questions. All right. Okay. So going back to the data, and there's actually an experiment going on here that I didn't mention. In the middle of this, so we had 278 seconds of video. In the middle of this video, the biologists are manipulating the worm, and what they are doing is giving it puffs of odor. Okay, so there's a 10-second periodic signal that's being given in the middle of the video, which is a on-off signal of odor. Okay, 5 seconds on, 5 seconds off. I think 9 of these repetitions, roughly in the middle of the video. And anyway, we'll kind of end up seeing that. And actually, if you look at the data carefully, you seem to notice that even just with the naked eye. Okay, so, all right, so our point cloud is in 720 dimensions because we have 72 neurons, and our window length is 10. Okay, so as we sweep across, we end up with, there were 278 columns, we get 269 windows of length 10 as we go all the way across. Okay, so this gives us a point cloud here, and it's of course ordered, but for the purposes of persistent homology, we just think of it as an unordered collection of points. We build the Vietoris-Rips complex from that, and we compute its persistent homology in degree 1 because we're looking for periodic behavior. All right. This is what it looks like, and nicely we have kind of 2 dots in our persistence diagram that are far away from the diagonal, so kind of 2 highly persistent loops in this high dimensional point cloud, and we can somehow try to visualize that by projecting it down to 2 dimensions. The question is, so a question from an expert in the front row was, when we are looking at this point cloud, when we turn that into the Vietoris-Rips complex, we're computing pairwise distances, and So of course there's a question of what distance you're using in R 720. And I said it was in Euclidean space, so if I was being completely corrected by my terminology, I would be using Euclidean distance. But we'll see later on that actually we don't end up doing that. But let me save that for later. Okay, so the big question for us, and of course it took us a while to get to this question of trying to understand what the biologists were interested in, what their data was, and then what our tools could conceivably do. And of course we didn't actually have anything that we could do kind of off the shelf that would answer things, because that maybe would have been nice, but maybe less interesting than actually having to come up with some new ideas, is this following question. How can topology help us identify neurons most responsible for driving these global cyclic brain dynamics? Okay, so there's some sort of quasi-periodic dynamics going on here. There's a lot of neurons. Can we kind of reduce that list of 72 down to something smaller and try to say something about what's going on here using applied topology? Okay, so there may be different ways of doing this, but this was our idea of how to do this. Our idea was to rescale neuron activations and recompute persistent homology. Okay, so let me say more about that. So here are 3 neurons and their time series and 2 different scalings of them. Okay, so I think if you look carefully, you'll see the 3rd neuron is scaled the same in both cases, but the first 2 neurons are scaled differently. And then if I combine those, we get 2 different persistence diagrams. And so in our setting, the persistence diagrams we had initially where all the neurons are weighted equally is this one here. And the question is, can we rescale the neural activations to increase the lifetimes of these 2 dots? Okay, so to make the intervals of these interval modules or bars bigger, or to increase the distance to the diagonal of these dots on the persistence diagrams. Okay, we want to increase the persistence of the 2 most persistent features in H1 of persistent homology. Okay, and so we had something very mathematical that we could do, and furthermore we have an interpretation that we can give to our scientific collaborators, which is that hopefully that the neurons that are scaled more highly after this optimization process will, we hope, would be kind of contributing more strongly to the global dynamics. Yes?
And did the domain specialist decide which ones to scale, or like?
Well, you'll have to wait till the end of the talk maybe to get that. I haven't showed you how to do this yet, so this is going to be kind of this is our aspirational goal, and at the end of the talk you can kind of decide if, I mean, to really settle this will require more work, kind of like is ongoing and kind of goes beyond what I can talk about today. Okay. All right. So how do we do this? So of course, if we just multiply everything by 10, then the persistence goes up by 10. So we want to normalize somehow, and we probably don't want to turn any of these activations negative because that's biologically not reasonable. So kind of all of these activations, maybe we want to scale between 0 and 1, and then we want all of the scalings to add up to 1. Okay, so that just says that we want to scale things using a point in the standard geometric simplex. All right, so already mathematically this is nice. We have a nice parameter space, which is the the standard geometric simplex, and we want to do optimization on that. Okay, so before I show you the details, let me give you one synthetic example to maybe hopefully convince you that this method seems to be reasonable and give nice answers. Okay, so this is our synthetic example. We have 10 sine waves here, all of which have amplitude 1, period 50, but some randomly chosen phase shift. And we've sampled them at 301, at the integers from 0 to 300. Okay, probably from the back of the room, this looks like continuous data, but hopefully at the front of the room, at least the people wearing glasses at the front of the room, can see that there's, this is discrete data. Okay, well, actually, I can't even tell that, but I can on my laptop, which has better resolution maybe than the screen does. Okay, so we have 301 dots tracing out a sine curve of various phase shifts. And, all right, so all very nice, idealized, everything should be beautiful here. All right, so let me do something awful. For 7 out of 10, I'm going to randomly permute. I'm going to shuffle all of those values. Okay, so pick an element of the symmetric group on 301 letters. Do that independently for 7 of these. And now we got this horrible time series, at least for 7 out of 10. And I'm going to do one other awful thing to the data. And to see that properly, I need to zoom out first. So that's the same data, just zoomed out a bit. And I'm going to add in IID Gaussian noise to each of these numbers with standard deviation twice the amplitude. Okay, so lots of noise. And hopefully now at this point you can't tell which ones are which. All right, of course we know the top 3 are the nice ones, but really they're all pretty terrible. Okay, so this is our data, and we're going to do a sliding window embedding on this, and to get a signal we need to use a really big window here, like 250. If we do that, there's one dot in the persistence diagram that's slightly further away from the diagonal than the rest of them. And we are now going to do an optimization to rescale the signals to push this dot away from the diagonal. Okay, so this is our algorithm, uses gradient descent. So we'll talk more about that later, but let me show you how it works here. So we start, so we're doing an optimization. The parameter space is in this case, the standard 9-dimensional simplex. And we are going to do gradient descent on that parameter space. And on the bottom here, you're going to see the updated persistence diagrams as we go through gradient descent. So it turns out that we can compute gradients of the total persistence, or the persistence of the most persistent dot in the persistence diagram. Okay, that's what this is here. So this is a functional on persistence diagrams. Just returns this one number, and it turns out we can compute its gradient. Okay, so we do gradient descent, and we're just going to do that a bunch of times. Okay, so over here I'm going to show you, this is how the update of the persistence diagram happens as we do this gradient descent. So of course we're not the first people to have done this. Lots of people have studied this, and in fact this kind of fits in very nicely with machine learning pipelines, and there's Python libraries that do this very quickly. And our emphasis is not to do the kind of understand the optimization and make it as fast as possible, but really this kind of interpretation that we're hoping is useful to in these types of applications. All right, so let me show you again. So we have this video. This is a kind of a path in the space of persistence diagrams. And on the middle here is something interesting. We're doing gradient descent on the 9-dimensional simplex, which you might imagine we have no hope of visualizing. But actually in the middle we have here a visualization of the path. Okay, so if you've never seen things in 9 dimensions before, here you can visualize 9 dimensions. All right, I always have arguments with my wife about whether or not we can see things in 4 dimensions, and I say yes. Here we see things in 9 dimensions, okay? On the left side we have the barycenter of the 9 simplex, and as we step through the simplex here, just using this color scheme, we end up at a point on the right-hand side in the 9 simplex. And the numbers there are these numbers here, and the first 3 numbers give us 80% of the kind of total scaling.
I'm sorry.
Okay.
What do the colors represent?
So the colors, so this is a heat map. The lighter it is, the bigger the scalar value. All the scalars are between 0 and 1, so you just need to pick your favorite color map that goes between one color and another. In this case, I don't know, purple and yellow or something.
That corresponds to closer to the other corner of the stacks.
Exactly. So let me repeat that. So at the end, we are at a point in the 9 simplex that is close to the 2-dimensional face, which is a triangle, which is the convex hull of the 3 vertices corresponding to the first 3 signals. Okay, yes?
You could try this method against some other methods like exponential smoothing of all of them and then trying to give the data from that? Or is it the only way you can get this particular 3 patterns?
That sounds interesting. I think we should talk about it after the talk. Yeah, I don't quite understand exactly what you're asking for. Yeah. Yes.
Did you also try? I mean, this point is just a bit more persistent than the others. Did you try with the second most persistent point?
It turns out it's the right one in this case. Yeah. So. In this example, we pushed the technique kind of as far as we could, and that's why the analysis here is somewhat delicate. But if you only apply half as much noise, then you'll see that you'll get one point that's quite a bit further than all the other ones, and then it's kind of the obvious one to choose. Yeah, so you could pick a different function to optimize, which would be like this. Yeah, and then I think things wouldn't necessarily work out. Yeah. So I should say the functional doesn't pick a particular dot in the persistence diagram. It just takes the maximal persistence across all of them. Okay, so I'm not quite sure how we would do exactly what you're asking. All right, great questions. Okay, all right, good. I'm about halfway through my talk, maybe a little further. So let me say a little bit more about the theory. So of course, we need some mathematical notation. Okay, we have a time series, and I'm going to use kind of statistical terminology here. We have using n and p here. I think this is the common letters used in statistics here. So we have n samples in p dimensions, so p multivariate data, and we have n observations. And we're rescaling them, as I said, using a point in this geometric p-1 simplex. And let me denote a rescaled version of the data by Vx here. So V is the vector in the simplex, X is my time series. So Vx is the rescaled time series. So just for each of the for each of the data points in the time series, we're rescaling the jth coordinate by Vj. Okay. And now some other notation. Capital phi of V is going to be the persistence diagram of the Vietoris-Scripps complex of the sliding window embedding of this scaled time series, rescaled time series. Okay. And then the additional ingredient I need is some functional on persistence diagrams, so something that turns persistence diagrams into a real number. This could be something like maximal persistence in the previous case, or total persistence. And we'll see in the biological example, it'll be the persistence, total persistence of the two longest bars, or two most persistent points. Okay. And then we have this optimization problem. Okay, so maximize this function for points in the geometric simplex. Okay, now as mathematicians, we're not content to solve a problem. What we want to do is find a method that works, and then find the most general class of problems that method works for. And then that is what we end up doing. And that also kind of ends up clarifying the notation, maybe to moving things into a more general setting. So to do that, we want to analyze sliding window embeddings, and this will end up coming back to the question that Michael asked earlier on. We're going to do something. So Michael Kerber's question was, what norm are we using for our sliding window embeddings? For the Vietoris-Scripps complex of the sliding window embeddings. And we're going to use the metric induced by the one norm. Okay. This ends up being crucial for the analysis, and it's also reasonable in the sense that we have a vector space with a basis that's given to us. Okay, each of the neurons or each of the time series is giving us a basis. So choosing kind of a non-Euclidean norm is reasonable, and we're going to choose the one norm. And because of that, we get this kind of nice lemma, which is that the distance matrix that we get for our sliding window embedding is the sum of the distance matrices that we would have got had we applied the same method just to each component one at a time. Okay, so this is kind of directly coming from the fact that we used the one norm. Okay, and because of this choice, we now see that something simple is going on here. And now we can abstract away the sliding window embeddings and just work with convex combinations of matrices. Okay, so that's going to be our generalization. So we have distance matrices now. We have a collection of p distance matrices. So in terms of time series, one for each signal, one for each component. And we have the same setup now. So we're looking for a point in the p-1 dimensional geometric simplex. And we're going to look for the corresponding convex combination that maximizes some function. Okay, so that's our generalization. Actually, that's our first generalization. I'm going to go one step further. And to do that, I need a bit of notation. And so, of course, people who have worked with TDA, which is, I think, most of the room, know that when we have a distance matrix, we want to turn that into a homology computation. We build something called the Vietoris-Scripps complex out of that. What is that? Well, that is a filtration of a certain simplex. Okay? So in fact, it's the simplex if we have a m by m distance matrix. It is a filtration on the m-1 dimensional, in this case, combinatorial simplex, not the geometric one. So this is what I have here. So this is what's usually used in TDA. I'm going to do something slightly different, which I don't know if other people, I haven't seen other people do this, but I'm going to advocate for this. I think it's a nice thing to do, which is just to add one more set to the m-1 dimensional simplex, combinatorial simplex, which is to add the empty set. Okay, so not just to use non-empty subsets, which are usually called simplices, but let's add the empty set. So it's a -1 dimensional simplex. And now when we take homology of that, it corresponds to taking reduced homology of the simplex. Okay, so we'll see that that has kind of a nice consequence. And one bit of language, let me call a weight on this augmented combinatorial simplex to just be an order-preserving map. So we're just assigning numbers to all the simplices so that no simplex appears before any of its faces. Okay, and the weight language is coming from graph theory, where people talk about edge-weighted graphs, for example, or more generally edge and vertex-weighted graphs. Okay, all right, so since this abstract simplicial complex is contractible, the homology of the augmented combinatorial simplex is 0. Okay, the 0-dimensional case is not a special case. Everything disappears. And the nice consequence of this is that all of the simplices are paired. Okay, so the analysis ends up being kind of nicer. All right, so this is the optimization problem that we ended up working with, is that we have a collection of p weights of this augmented combinatorial simplex, and we're optimizing some functional on persistence diagrams of the convex combination of these weights. All right. I think in the interest of time, I might skip over this. Let me just say briefly that the nuts and bolts of everything that's happening here is that as we do the optimization, we get a one-parameter family of persistence diagrams. This is sometimes called a vineyard. It goes back to Dmitry Morozov's PhD thesis, and there's a beautiful corresponding paper by Cohensteiner, Adelsbrunner, and Morozov. And anyway, we studied this kind of very carefully in our case, and we showed that when we do the gradient descent, these vineyards are piecewise linear, and a straightforward consequence of that is that all the persistence diagrams are stable. Okay, so this is essentially the proof of Scraba and Turner and the earlier proof of Cohensteiner, Adelsbrunner, and Morozov. Okay. All right. So it is a math talk. There should be one theorem here. So at least, so there's a theorem that when we do piecewise linear gradient descent, then the values of these functionals will be stable. All right. So okay, let's get back to the motivating example. And all right, here's a picture I was looking for earlier. It wasn't there, but now we. So we have these two dots in the persistence diagram, and as we step through the time series here, if we look at the two-dimensional projection of the sliding window embedding, okay, this is a picture of the 2D projection using PCA of this 720 dimensional point cloud. And it's ordered, actually, because our windows are ordered. We don't use that ordering in the persistent homology, but we can use it to help understand what's going on. And if we color it kind of from purple to yellow, you see that the point cloud travels around like this, goes around here, and then comes back. Okay, so there's two loops, and actually the second one here corresponds to kind of the middle of the video where the experimental perturbation happens. And then this other loop is actually, you can kind of see it here, it's this part here. And then there's two paths that somewhat coincide, and those are these parts and this part here. All right, so we do our gradient descent. So another part of the theory that I didn't have time to get into is that there's actually a partition of the geometric simplex, and for many of these choices of functionals, and the functional we're using here is the sum of the lengths of the two longest bars, the sum of the distances from the two dots in the persistence diagram to the diagonal. That functional, its gradient is piecewise constant on some partition of the n simplex. Okay, so computing these gradients is easy. So it's piecewise constant, so you just compute that gradient, you flow some direction, some amount. We just did a fixed amount. And then, all right, I think I have a video here. Yes, okay, excellent. So this is what happens here. So on the bottom here, we're doing gradient descent to, in some sense, push the two most persistent dots away from the diagonal. And this is the one parameter family of persistence diagrams that we get. And on the top, we have a visualization of the corresponding path in the 71 dimensional simplex, okay? And, okay, so just as before with the synthetic example, we're starting at the very center, all the components are weighted equally, and then at the end they aren't. And now our hope is that it's something interpretable. And so first of all, many of these end up being very close to zero. So 32 out of the 72 have values or scalings bigger than 0.001. All right. And these are those 32 neurons. I've written down their names. Okay, so for those of you in the audience who know your neurons and C. elegans, okay, so of course I don't really know these, but my postdoc, my former postdoc John Bush, who did most of this work and all of the computations, talked with Hong and her students and learned about what all of these neurons were responsible for, or what is known about them. And then the short version of the story is that the neurons that are on the top of our list are ones that are sensible to have expected to have shown up in this analysis. Okay, the top one here, RIBL, is a head interneuron known to integrate inputs from sensory signals and contribute to local motor regulation. Okay, so similarly, the other ones are kind of motor neurons involved in head movement, because when the worm is searching. That's kind of one of a quasi-periodic behavior you'd expect to see. And also interestingly, yeah, we have a chemosensory neuron because odors, puffs are appearing, so this is things that we would expect. All right, so yeah, so we have a mathematical method. I think it's broadly applicable. We applied it to our data, and we got reasonable answers. And if you want to see more about the details, there's a paper on the archive, and the latest version just appeared today. So please check that out. And that's the end of my talk. Thank you.